Hey everyone, welcome back to DIY Biotech. My name is Nick. I have a PhD in genetic engineering, and today I'm very excited to share this idea of artificial photosynthesis with you. So we can now more efficiently do photosynthesis than plants. And this is proposed by a research group that was across the hall from mine, and I know many of the authors of this paper. And so hopefully I can provide some insight sort of beyond the paper, which might be helpful. So I want to start with this graphic right here showing exactly what we mean by artificial photosynthesis. So traditionally we have plants, right? The plants are going to take in some solar energy and convert it to food for us. And with traditional agriculture, as you can see right here, we get about less than 1% biomass for the energy that the sun puts in. And this can be from losses, and we'll look at another graphic in a second, but this can be losses from, certain wavelengths of light aren't absorbed by the plants, the actual efficiency of the photosynthetic process can be lower, whatever. The total efficiency is less than 4%. With this proposed artificial photosynthesis system, instead of having plants, grow naturally, we can instead have photovoltaics, so we can have these solar panels that capture more energy from the sun. So with photosynthesis or with solar panels, we only lose about 80% of energy from the sun. And then we use electricity to fix carbon dioxide. So we can take carbon dioxide out of the atmosphere, just like plants do, right? Plants using the sun's energy to capture carbon dioxide and turn it into food potentially for us, right? They're turning the carbon in carbon dioxide into the carbon in our corn and our apples, right? That we're eating. So we're using electricity from photovoltaics to convert carbon dioxide in our atmosphere into small carbon products like acetate. So acetate is just two carbons and some other stuff going on. It's basically neutralized vinegar, right? And then we can use the acetate to grow our food products instead. So normally, if we have a plant, it's making its own sugars, right? The plant is taking energy from the sun, taking CO2, and turning it into sugars and other carbon products that we're going to end up eating. If instead we want to grow other food products like mushrooms, for example, we have to feed the mushrooms some other carbon product. So, like waste. So wood, we could feed them wood. So we could grow trees and we could take the wood and then turn it into mushrooms. But instead we could feed those mushrooms acetate that we pull straight out of the atmosphere through carbon dioxide. And it turns out that this process of artificial photosynthesis, since photovoltaics are so much more efficient than plants, we can actually get about 4% biomass from the energy that we get from the sun. So this artificial photosynthesis system right now is 4 times more efficient than plants. And so what does this mean for us? So I want to dig into this paper first. This is a newer paper from 2024. You can see this author, Feng Zhao. I've met him in person a couple weeks ago. This is early November when this is being recorded. But I went to a chemical engineering conference recently. I met Feng Zhao in person. Robert Jinkerson is the professor that works across the hall from my lab. He was also on my dissertation committee, so I know him decently well. Marcus I know decently well as well. We're on a paper together. And so, I actually texted Marcus a couple questions about this paper, which we'll get to in a minute. But this paper is more of an overview of the possibilities of artificial photosynthesis, implementing the current efficiencies of the whole process. The main paper that we're going to look at is from 2022, and so there's actually a few small improvements that have been made in the process. So I think this paper is a good starting point because it gives us a lot of perspective of how powerful this artificial photosynthesis technology could be. So this first sentence here is fantastic. Adopting a food system based entirely on electro-agriculture could reduce United States agricultural land use by 88%. 88% less land. And that's right now with the current efficiencies of this process, right? And As far as land use is concerned, nearly half of the world's habitable land is being used for agricultural purposes. Half of the land that we can inhabit is used for agriculture. That's insane. So let's look at this picture down here then to visualize this. You can see this is how much land the US uses for agriculture. It is insane. And with the current specs of artificial photosynthesis, we could cut this down by 88%. So all of this light green area could instead be reforested, or people could live on that land, right? This is fantastic. This is a really, really optimistic future. And I highlighted one more thing here I wanted to talk about. With this system, we could produce food year-round anywhere in the world. So instead of just freeing up this land that we can live on, instead we could move all of our food production to like the Sahara Desert, for example, or the Mojave Desert in Western United States. So instead of using land that could be reforested, or instead of using land that could have forests and this wonderful wildlife or places that we could live on, or national parks, we can instead grow food in areas that are less utilized, or can be less utilized by other industries and by people in general. This is really fantastic stuff. So let's look at the main paper I want to talk about today. It's titled "A Hybrid Inorganic Biological Artificial Photosynthesis System for Energy Efficient Food Production," which is exactly what we're talking about, right? Again, the main professor on this paper is Robert Jinkerson. He was on my committee. Feng Zhao, he does the electrocatalysis stuff. Robert is in the chemical engineering department at my university, but he does a lot of plant stuff as well, which I think is always cool to have that multidisciplinary touch. Dang, I know personally, he's a cool guy. He's a year below me. Liz, I knew decently well. She's now doing postdoctoral work. She got her PhD already. Really cool stuff with algae. And then Marcus, again, I know he's a year ahead of me, and he just defended his PhD just last week. So congrats, doctor. So let's go through this paper and see what 2 years ago was the most efficient way. And it's pretty close to what the most efficient artificial photosynthesis is now. So what exactly do we mean by artificial photosynthesis? What are the details? Where are we getting these numbers from, right? So the Feng Zhao group has worked for a while on producing a system to convert carbon dioxide into these, small carbon molecules like acetate. They've tried a bunch of different catalysts and different reaction conditions, and they've come up with this method that has 57% carbon selectivity to acetate. So 57% of the carbons from the carbon dioxide end up assimilating into acetate, which is fantastic. Then we can feed this acetate to other organisms that can be food for us. So heterotrophic means that they're using some carbon source. They have to have some carbon source input, whereas autotrophic would mean they can produce their own carbon, right? So plants are photoautotrophic. They use light to make their own carbon sources, whereas heterotrophic, they need to be fed a carbon source. So we can force plants and algae to use a carbon source instead, and so we can grow them in a heterotrophic way. So here we're going to talk about heterotrophic cultivation of yeast, mushroom-producing fungus, which is always fun, photosynthetic green algae, in the dark without inputs from biological photosynthesis. So in the dark, we're growing algae in the dark, right? You think it's a photosynthetic organism, it is, but we can also grow it heterotrophically. Algae is really good at doing this. Regular plants are less good at doing this, and we'll talk about that more. So they showed that from crop plants, so you can think of like corn or beans, they tried a few different crop plants. They found that acetate is incorporated into the plant. So we can't yet grow plants completely in the dark just on acetate. Maybe that's the future dream. But from this paper, they've shown that the plants can at least take in some acetate and convert it into parts of the plant, which is cool. So let's dig in. Most crop plants can convert sunlight and CO2 into plant biomass at an energy conversion efficiency of only 1% or less. So this is energy conversion efficiency. So given, a square foot of land that a plant can grow on, only 1% of the light energy that comes in is actually converted to our food, which is terrible. Hopefully we can bump that number up. Okay, so we're talking about the catalyst already. I really want this reactor to be something you could build at home. So you could build, the CO2 to acetate reactor at home and play around with it. I think you could figure it out, but there are a few barriers to entry. One of them is this nanostructured copper catalyst. I'm not sure how you make that. Maybe one day I'll dig into it. Leave a comment below if you want me to dig more into the electrocatalysis side of things. It's a little bit above my head, admittedly, but it is really cool stuff. So I would love to learn more if people are interested in it. But one of the big breakthroughs here was this nanostructured copper catalyst. In this first results section, they're going over the electrocatalysis stuff. So they also talk about this iridium oxide catalyst for the reduction of CO2 to acetate. And from this figure here, you can see they actually use 2 electrocatalysis cells. The first one converts carbon dioxide into carbon monoxide, and then the carbon monoxide can be converted to acetate. So we have to have that first step, and so they're using iridium oxide as the catalyst, as the main catalyst for that step, or rather for the anode. They made some reactor that made a small amount of acetate, but the percentage of acetate relative to the electrolyte, so the electrolyte in this case is potassium carbonate, I believe. We'll look at it in a second. But the ratio of that acetate to the electrolyte was not very good. So they're producing a very tiny bit of acetate, and then they have this really high salt concentration. And they found that growing algae on this high salt concentration, low carbon concentration, didn't work very well. So they wanted to maximize the ratio of acetate to electrolyte in order to give it to plants or other food sources. So one of the big improvements that they made is adding this carbon dioxide scrubber. They found that this second electrolyzer was not very efficient if there was any CO2 getting into it. So they added this scrubber, and they showed they got really good conversion to acetate mainly. And as a side note here, they are making other small carbon molecules. So they're making ethylene, propionate, propanol, and ethanol. And these carbon sources could be used by organisms as well, but those pathways are either less efficient or these smaller carbon molecules are toxic to certain organisms. And so acetate is just inherently less toxic. The metabolism is much easier. We can convert the acetate directly into this central metabolite called acetyl-CoA by just one enzymatic step, right? So instead of a long step with a lot of side products and stuff, if you were trying to break down propionate or ethanol or something like that, acetate is converted directly into this central carbon metabolite that the cell can use readily. All right, so the exciting part here is that they're growing food on this simulated effluent from the reactor. So they ended up getting a reactor effluent that contained almost 0.7 molar of acetate, which is a lot. It's a lot of acetate. It's something like 30 grams per liter, I think is the number, somewhere around there, maybe 40. And then, so their electrolyte was actually potassium hydroxide. So you can imagine potassium hydroxide may not be the best chemical to feed microorganisms or organisms in general, and 1 molar is a lot. 1 molar is a really high concentration. But even with that high concentration of potassium hydroxide, they were able to grow algae in the dark, feeding them just this effluent. And this simulated effluent also contains these side products, the ethylene, the propionate, the propanol, and the ethanol. So this is a very realistic scenario. They're taking basically the same thing that's coming out of the electrolyzer and feeding it to organisms. Fantastic. So first they showed this with algae growing in the dark. So this is heterotrophic, the algae must be eating the acetate and converting it into biomass. So they're converting it into their cells, which is going to, those cells are going to consist of proteins and carbohydrates, which are things that we can eat. And sure enough, this species of algae, Chlamydomonas, something like that, clammy for short, is already used to supplement some foods, and it's being looked at as a more affordable and more economically viable food source compared to growing animals or plants. So they also tested, this is, let's see, panel B is showing the clammy cultures growing in the dark. So this is acetate versus the effluent. So you can see in the pink, this is just acetate, and then in the green is the effluent containing the side products, right? And you can see there's no significant difference in the dry weight after the growth experiments, and the growth curve looks about the same between the two. So those other products that are happening in the electrolyzer effluent aren't significantly limiting the growth of algae, which is fantastic. So this algae system is the one that's working really well right now. You could do this at home. Anybody could do this in another lab. You get a strain of this clammy algae, or probably any algae, and grow it on acetate or grow it on this simulated effluent, and it will grow fine, right? Very cool stuff. They also showed this in panels D and E with Saccharomyces cerevisiae yeast. So Saccharomyces cerevisiae is the yeast that we bake bread and brew beer with. Of course, you could go out and buy Saccharomyces cerevisiae right now and buy vinegar or sodium acetate, vinegar and neutralize it with sodium hydroxide or something like that, or just buy sodium acetate or potassium acetate or something like that, one of those salts, make media with the acetate, and then grow Saccharomyces cerevisiae on it. And potentially that vinegar or the acetate that you're purchasing could be cheaper than growing your yeast on glucose, which is really cool stuff. So acetate is already potentially a cheaper carbon source than glucose. So normally we would just grow yeast on sugar. And this brings me to the collaboration that I had with the Jinkerson lab. So Robert Jinkerson is here. Here's Marcus. Him and I worked together on this project in the beginnings of it. And this is a paper that I've already gone over on my channel, but I'm the first author on this paper, so this is my baby. This is my paper, so I better know all the details of it. And so you can see here that we were growing this yeast called Yeroia lipolytica on 0.5 molar acetate. So again, we go back to the this paper that we're focusing on, and they're growing on 0.7 molar acetate. So for Saccharomyces cerevisiae here, they're growing with 0.75 molar acetate, which is really high. For the paper that I put together, we used 0.5 molar acetate. So we were evolving a strain of Yeroia lipolytica, this oleaginous yeast that makes a lot of lipids. We're trying to identify gene mutations that improve growth on acetate, potentially for something like artificial photosynthesis. And so these are growth curves from growing in 0.5 molar acetate from different mutants. Basically, the Yeroia lipolytica yeast also grows well on acetate. And from this paper here, let's see if I can find it, the algae produced 0.28 grams of algae per gram of acetate, so 28% efficient. And I just texted Marcus and I was talking about this number, and he said it's actually closer to 50% now. So 50% of the acetate that you feed algae is going to be converted into algae, which is incredible. And then for Saccharomyces cerevisiae, they reported 0.19 grams of yeast per gram of acetate fed, which is a little bit lower, but Marcus said this number is also actually much higher, closer to 50% as well. And then when I grew Yeroia lipolytica, these were shake flask experiments. So we had a flask with 250 milliliters of, it was 50 milliliters of Yeroia lipolytica growing in just acetate. So there's other salts and stuff in the media, but it was just acetate. And then we grew them for a while, and then I took a sample of the cells, spun them down, dried them out, and weighed the cells. And what I found was, this is the milligrams of dry weight per 10 milliliters of culture. But if you do the math on all of this and convert from moles to mass, whatever, I got to about 25%. So in this context, 0.25 grams of Uroa lipolytica yeast for every 1 gram of acetate fed. So these are crazy high efficiencies. So, this food that we're feeding the yeast and the algae is very efficiently being converted into the yeast and the algae, which is really, really good. So let's go back to where we were. They also looked at the fruiting mushrooms. So they're looking at oyster mushrooms and enoki mushrooms right here. And in this one, it's a little bit more disappointing. They had to dilute the simulated effluent by about 10 times. So they're using 0.07 molar acetate instead of 0.7 molar acetate. And that's what this 0.5% converts to. So if they did 1%, which would be about 0.12, 0.14, somewhere around there, molar acetate, the fungus didn't grow very well. So yes, we can feed acetate to various fruiting fungi, but from at least preliminary data, they don't grow very well on it. They are growing on it, they are utilizing it because that was the only carbon source here, but it's toxic at higher levels. And the authors of this paper speculate that the main problem here is propionate, I believe. I don't exactly remember where that part was, but they say that propionate, I believe it was propionate, is an antifungal compound. And so having propionate in the reactor effluent is really bad for the filamentous fungi. And so potentially if you could engineer the electrolyzer to have less of the propionate, then maybe you would have more robust growth of these filamentous fungi. Similar results were seen for plants. So why algae, first of all? So this paragraph is really good, and I touched on this earlier as well, but clammy is added to processed foods for protein fortification and has been shown to have a positive effect on human gastrointestinal health. Really cool. Algae can produce large amounts of starch, protein, and oil, like I mentioned earlier. Clammy grown heterotrophically can produce over 1 gram of starch, 1 gram of protein, and almost a gram of lipids per liter per day. So a little bit slow, but pretty good if this is heterotrophic. We don't have to depend on light, which is the main problem with algae, right? You need a lot of area for the algae to grow to produce products that you want. And we don't need to be producing food with algae. We could also be producing valuable biochemicals. And so there's some potential there. Anyway, I was trying to talk about plants. With plants, from what I understand, they were only able to feed plants pretty low concentrations of acetate, and the amount of acetate that actually gets assimilated or put into the structure of the plants is pretty low. So they did this really cool technology called carbon-13 labeling. So they essentially got a version of acetate that, instead of made with the sort of naturally occurring C12 carbon, they have this labeled C13 carbon. So the acetate is made from C13 carbon, and so when the plant grows on the C13 acetate, the carbon that has that C13 labeling ends up getting incorporated in other molecules in the plant. And there are methods to see C13 containing molecules, and that's what this heat map is showing here. So we have darker red, meaning there's more of these molecules right in the rows in these plant or conditions that contain carbon-13. So this is basically showing how much acetate is being assimilated and into what molecule. So for example, we can see in tomato, in this column, that there's a lot of this acetate being assimilated to aspartate, this amino acid, and serine, as well as disaccharides. So think sucrose is a disaccharide, right? And less of the acetate is being converted to succinate in tomato, for example. And then the sort of best performing one that seems interesting to me, or that I wanted to point out, is this dark grown lettuce callus. So if you give acetate, just acetate, not the effluent, just acetate to lettuce callus grown in the dark. So lettuce callus is, you take a tissue sample of lettuce, put it in special media that makes it grow in basically a cancerous state. Then you can grow that in the dark on acetate and see how much of that acetate gets assimilated into what product. And for whatever reason, in this experiment, it made a lot of aspartate. So this was the only one grown completely in the dark. There's a really good SciShow video on this paper, actually, and I was really excited when it came out. And I was like, oh yeah, I know the group that did that paper. I know that paper. And they mentioned in the video somewhere like, oh yeah, you know, the authors are saying that we can grow plants on acetate, but they did all their experiments in the light. There was a good reason they were all done in the light, because the plants can't grow completely in the dark only on acetate. That's just not possible, at least with these plants. They're the authors of this paper are in the process of designing plants that can more efficiently grow in just acetate and no light. But I think there's a lot of engineering that we would have to do to get to that stage. To summarize this plant section, I think algae and yeast are very promising candidates to convert this electrolytically produced acetate directly into food. And potentially fruiting fungi could be used as well, but there was this issue with propionate, and plants are much further away from assimilating acetate. However, potentially we could have hydroponic systems that contain small levels of acetate, which would maybe reduce the amount of light that you need to grow these plants by, I don't know, 10% or something. And that could be a huge energy savings, right? Because converting the sun's energy or other renewable sources of energy into light for plants is pretty inefficient as well. Those lighting systems produce a lot of heat, and they only convert about 20 or 30% of the energy into light for the plants. Correct me if I'm wrong in the comments, but it's something like that. But this algae, is converting 99% of the acetate into stuff, and 28% of it, or what Marcus is saying, closer to 50% of it, into actual algal biomass. And similar numbers we've seen for yeast, which is really, really cool. And so we have this picture again, or a similar picture, comparing biological photosynthesis to artificial photosynthesis. So again, a lot of this solar energy is outside of the spectrum that chloroplasts and chlorophyll are going to absorb, right? About 51% of it is just impossible for these plants to absorb. And then we have light interception efficiency, so how the leaves are angled potentially, and then the photosynthetic conversion efficiency. So from that light that the plants actually get, most of it is not utilized efficiently. So we end up with plants around 1% biomass, whereas again with artificial photosynthesis, we're converting about 4% of the energy from the sun to biomass in plants. Really, really cool stuff. And these numbers are based on commercially available solar panels. So let's see where the potential is now, like what we can improve upon, and where the future is going, right? So this graph here, I think, demonstrates really well what we can do in the future. So right now we have about 4% electroagriculture efficiency, or artificial photosynthesis efficiency. Down here is 1% traditional agriculture. Solar panels are going to get more efficient. Within the next decade, potentially, we have solar panels that are around 40% efficient commercially available. And so that's going to provide a huge benefit. The electrocatalysis efficiency is also going to improve somewhat, or is projected to improve. And then the cultivation of these organisms is also projected to improve. So potentially, in the next decade or two, we could have a system that's 10 times more efficient than traditional agriculture. Really, really, really cool stuff here. And this is, again, sort of summarizing what I've already said, but basically, yeast, algae, and mushrooms, we can grow on acetate pretty well. We have some pieces in progress, and in the future, there are other plants that we may want to grow on acetate as well. And let's see. Oh yeah, so here's the section where they say propionate is an antifungal, and then with the C13 labeling for the plants, they were only feeding them 2 millimolar of acetate. 2 millimolar. This is a really, really, really small amount. Before we're looking at hundreds of millimolars feeding to algae and yeast. So for plants, maybe we're a little bit further away from this cool new future, but for algae and yeast, we're already there. We can do it right now. And then finally, I wanted to talk a little bit about, if you were to build this electrolysis system at home, this is the method that you should look at here. So here's the electrolysis method. It has a bunch of details on the anode and cathode and flow rates that they used. They're using, from what I understand, a pure carbon dioxide gas flow at 7 milliliters per minute. And this is potentially, less ideal than we would think. So, plants can take really low concentration carbon dioxide, something like 100 parts per million, right? Something small, really small concentrations of carbon dioxide in our atmosphere, and convert them into plant, right? With this method, you have to feed the electrolyzer pure carbon dioxide, or if you have a source of carbon monoxide, you could use that as well. You can get these sources, but you can't just have this electrolyzer sitting in a field somewhere. Ideally, you would have some industrial process that produces a lot of carbon dioxide already, and then you can convert that to acetate. And a lot of these industrial processes are producing carbon dioxide and carbon monoxide, and this process is even more efficient with carbon monoxide. So just some things to consider. If you're planning on doing this or trying to make this electrolyzer, please let me know in the comments below, or if you have experience with these kinds of things. That would be really, really cool to see somebody try to build this at home. If you want more details about this electrolyzer or how to put it together, let me know in the comments below. And if you want to see me try to grow different organisms on acetate, I think that would be really fun. I could do that pretty easily. I think I have the tools to do that if I need to. Let me know in the comments below. But with that being said, thank you so much for watching. Let me know any papers that you want me to cover next in the comments below. Subscribe, become a channel member. If you really want to support the channel, that's the best way to support me, is by becoming a channel member. Thank you so much for watching. Bye.